Once again, welcome back, students. Continuation to the microbial culture and its application. Today, we will discuss about the microbial nutrition and culture technique. Again, I would like to talk over that topic. As the animal cells, as the plant cells require nutrient medium for the culturing purposes, likewise all these microbes also required some conditions, that is the physical condition, chemical condition, for their continuous profuse growth. So continuation to that, there is basically two conditions are required, because before going to culture any particular microorganism, we must know that how do we culture a microorganism and what is the salient feature of that microorganism, where it lives, or which is the optimal temperature, optimal pH, optimal pressure required for the proper culturing of that particular microorganism. So keeping this consideration, here we are taking two basic things, two basic requirement. Number one is called physical requirement and number two is called chemical requirement. So these microorganisms is this microorganism is needing this physical requirement, and you know the physical requirement all over the earth is varying. It is not equal in everywhere. If we are discussing about the Antarctica, so you know that is the sub-zero. It means that whole throughout the year the temperature goes zero. And if we are asking about the hydrothermal vents, that is hot water spring, their temperature is more than 80 degrees centigrade. So similarly, as we move down to ocean, then the pressure will be increases. So because these microorganisms are ubiquitous, that is everywhere they are present, where you cannot expect the possibility of the, and even that condition these microorganisms thrive well, that is they survive well because of presence of their own metabolic system, because of presence of their own chemicals, and that chemical provide power to microorganism live in that suitable condition. You also know about the archaebacteria, and these archaebacteria are mostly found in harsh situation where life is not possible, and they are found in the Dead Sea. You cannot consider what is the pH of Dead Sea, or they are highly salty. So they are present in the salt lake, they are in the acidophilic medium where the highly acidic nature is there. And last one is, you know, the methanogenous bacteria or methanococcus bacteria. So these archaebacteria are also present in that methanogens producing gas, normally we call it the gobar gas production. So here this archaebacteria is surviving well because of their own unique properties that present in their plasma membrane. So keeping this, we would like to know that what is the basic requirement for the physical culturing of that microorganism. So first one is pH, temperature, pressure, and already I told all these are varies when we move from when we move from plain to deep ocean, or when we are on the hilly region. So there is a climatic changes occurs. Apart from that, if you ask about the hydrothermal vents in the deep ocean have very high temperature, they have very high temperature and they have high pressure also. Apart from that, there is alkaline ecosystem, acidic ecosystem and neutral ecosystem. So these are the unique feature of a microorganism. So we must know which one is required for the proper growth and culture of the microorganism. Similarly, important part that we call the chemical requirements, and that chemical requirements are starting from the macronutrients and micronutrients. We already studied about that. The first basic requirement is the use of elements. That is the chemical elements. We call it the chemical elements. And these chemical elements are categorized into the two groups that we call the macro element and the micro elements. What is macro means big, means it requires more. So these carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, these are the basic things. More than 17 nutrients, chemical elements are there which are required for the growth and culture of either plant cell, animal cell or the microorganism. So carbon, hydrogen, oxygen, nitrogen, phosphorus and sulfur, since they require in more quantity, though we call it the macro element. And on the other hand, there is some elements which require in less quantity or they require the little quantity, that we call the micronutrients. And for kind information, microelements are also known as trace element because they require quantity, and molybdenum, cobalt, zinc, etc. These are the trace elements. Now, nutrient medium. Nutrient medium is the medium on which we are growing or we are culturing the microorganisms. Since microorganisms are not able to create, not able to synthesize some desirable products for which is required or which is very essential for their growth. So we are here using nutrients medium, and these nutrients medium are categorized or they are divided into the two group, that is called the synthetic medium and the complex medium. Synthetic medium, we already studied about that in the tissue culture and the animal cell culture, because these synthetic medium have the chemical composition means precisely known chemical composition, that is the appropriate quantity is merged or it is included. So synthetic medium is also known as chemically defined medium because the chemical proportions are known. And for example, MS medium, niche medium and LS medium, this we have already studied about that medium. Now come to the complex medium. These complex medium do not have the known proportion of the chemical, so we call it the complex medium. And these complex medium, NB medium, NB stands for nutrient broth. Now the urea, LB, that is the urea broth, and there we are using the two words broth and the agar. So if we are using the liquid medium, so we use the technical term broth, and if we are using the agar, if we are using the solid medium, then we are using the suffix agar. If I am saying the example, suppose A and B, N stands for here nutrient, B stands for broth, that is it is a liquid medium. Now similarly urea broth, it is medium. If you want to change in the solidified medium, so here we write the urea agar and here nutrient agar. So this is the solidified medium or solid nutrient medium. Now this is the two physical state of nutrient medium. So physically nutrient medium are found in two forms, solid and liquid. So continuation to that, there are some of the important key points are required while we are considering about the nutrients medium. So first one is that it should give the maximum yield product or biomass per gram of substrate. It means the rate of production, that is biomass production. What is the biomass production? Biomass production is the total weight of total living weight contains of an organism is known as the. So while we are using this nutrient medium, so nutrient medium has power to yield good biomass production. Second one is that cause minimum problems during preparation of sterilization. Sterilization is a technique of removing the germs present culture vessel or the culture medium. So these nutrients medium have resistance power, or they have the power to defy the microorganisms, so that there is no need of requirement of less sterilization. Another one is that give minimum problems during production process, particularly aeration, agitation, extraction, purification of the products. If these nutrients medium are whatever the nutrient medium we using, so finally we want the large scale production of the products, so that large scale cannot be disturbed by the nutrient medium in their quality product production. So these are the some important points we are taking while we are preparing the culture medium. Now, what are the various sources of this nutrients medium? Let us see. The first one is the source of carbon. The source of carbon, as I told you, the carbon is the one of the basic things, basic elements required for the culture medium. So carbon source normally we are taking from the molasses, starch, cereal grains. Now nitrogen. Nitrogen we are taking from the ammonium salt, urea, corn steep liquor. These three things we are taking as a nutrient source. As a nitrogen source. And you must know that there is a one proportion we call the C/N ratio. C/N ratio. What is the C/N ratio? Stand for carbon and N stand for nitrogen. A particular there is specific ratio required for the growth of the either plant cell, animal cell, or the culture. C/N ratio. So carbon and nitrogen. Now the growth factor. You know, these microorganisms are not able to produce any kind of hormones. So we here providing vitamins and amino acid, that is for the growth purpose or the growth factors. Apart from that, energy sources. These energy sources is what here we are using the normally carbohydrate, lipids and protein as a source of energy. Now anti-foaming agents. What is the anti-foaming agent? Actually, when there is culturing process takes place in the vessels, what happened due to the metabolic reaction or due to the fermentation process, foams are very injurious, or they are very toxic, or they are able to not giving the suitable desired products. So we are using the anti-foams that suppress the formation of foaming. And the anti-foams which we are using here are silicones, fatty acid, olive oils. So at the time of culturing process, these anti-foaming agents are added in the nutrient medium or in the culture medium, so that it suppresses the foam formation. Now some other example about that. Energy source already we have done, anti-foaming already we have done. So I just told about this synthetic medium. Synthetic medium already told, that is the MS medium and LS medium. Once again, this synthetic medium have the known proportion of the chemical substance. On the other hand, this complex one, there is unknown proportion of chemical substance. So previously synthetic medium is also known as chemically defined medium. So this is about the microbial nutrition and culture technique. Once again I review that there is two categories of the physical requirements and the chemical requirement. These two things are required for the proper growth of a microorganism, because before going to culture the microorganism, we must know the basic things of the microorganisms, that where it lives, what condition is preferred, what is the optimal temperature required for the microorganism. So pH, temperature, pressure, etc. And you know that there is variation on the earth everywhere. And if you are asking about the hydrothermal vents, that is hot water springs. So here there is a very high temperature. It ranges between between the 80 to 100 degree centigrade or 80 to 90 degree centigrade where normal life is not possible. Apart from that, if you are asking about the Antarctica, so there is sub-zero temperature, there is a temperature always goes to zero. If asking the ecosystem may be alkaline, acidic, neutral, etc. like the salt lake or the Dead Sea. I just told about the archaebacteria, one of the unique example for that suitable condition. Now come to the chemical requirement. Chemical requirement, there is a requirement of the two things. First one is the microelements and macroelements. Macroelements are required in the large quantity, and carbon, hydrogen, oxygen, nitrogen, phosphorus and sulfur. And another one is the molybdenum, copper, zinc, nickel. It require in the least quantity, therefore we call it the trace element also. And apart from that, if we are going to that nutrient medium, so this nutrient medium is of two type here, synthetic medium and complex medium. Just I told about the synthetic medium is having the predefined the chemical composition, and complex medium do not have the appropriate proportion, that is their proportion is unknown. So we are using two state of the nutrient medium, solid and liquid. For the liquid we are using the technical term broth, and for the solid we are using the technical term agar as a suffix. Now anti-foaming agents, these anti-foaming agents which we are using here, silicon, olive oil and fatty acid. These foams disturb the production of the desired products. So anti-foaming agents are added here. Source of energy, lipid, carbohydrate and the proteins are the source of the energy. Now the growth factor, since there is no particular growth promoting substance by the microorganism, so we are applying here vitamin and the amino acid. So about the requirement, basic requirement for the culture of microorganism. Thanks for watching.